Oh, thank you very much, and good morning, everyone. So I'm with Zheng at the Princess Margaret Cancer Center and the University of Toronto, and what we focus on is creating multimodal, multifunctional nanoparticles for imaging and treating cancer. We work on small molecules as well, but these days we're mainly focused on creating nanoparticle platforms that are multifunctional, as I said, but also clinically translatable. And really, our vision is creating complexity in capabilities, but structural simplicity. So I want to talk about more of our approach to creating photoacoustic nanoparticles. And I'm going to start that talk not talking about photoacoustics, but talking about fluorescence for two reasons. One, we envision that a lot of the work that we do currently with fluorescence, whether that's preclinical or future clinical applications, we can do with photoacoustics as well. And the second reason is that in fluorescence, there's a very wide library of probes that are available that have many different functions, different capabilities, and so we can draw on that library to create our photoacoustic probes. So if you look at just fluorescence probes in general, there's two main classes. There's always-on fluorescence probes that can be targeted, and then there's ones that have some sort of responsivity to a biological interaction or sensors. And these are called activatable probes, and they either have an intensometric response or a ratiometric response. And the classical example is, of course, molecular beacons, protease or nucleic acid responsive beacons, that you can make them have either an intensometric response, normally through FRET from a fluorescence probe to a quencher molecule, or you can make them ratiometric by just switching out that quencher for another fluorophore. So I want to keep this idea of having something that's responsive to its environment or to some specific biological interaction, and how can we get that into a photoacoustic probe? And so molecular beacons are just one example. You can make these activatable fluorescence probes in nanoparticle systems, even just small molecules. But what they all have in common is these four different photophysical mechanisms for manipulating fluorescence. Most of them are quite simple, and you know them already. I'm not going to go through them. There's self-quenching, also called contact quenching, aggregation quenching, exciton coupling, which is a little more new of an approach. There's two different classes: J aggregation, which causes a red shift in the absorption and fluorescence spectrum, and H aggregation that causes a blue shift. And then also, of course, resonance energy transfer, commonly called FRET, and then photoinduced electron transfer. So the bottom two mechanisms are really specific to fluorescence, and they're not very useful for creating photoacoustic probes, because what we want is a change in how much fluorescence we're getting, not what wavelength. And exciton coupling specifically is really interesting in our perspective, because it allows you to change the ground state complex of your dyes. So you can change the actual spectrum, and you can change their photoacoustic spectrum that way. Most photoacoustic probes, though, of course, are currently focusing on self-quenching as the mechanism to produce a photoacoustic signal. So I'm going to go through three different probes that we have developed. One that's based on self-quenching, two that are based on J aggregation, and one of them has actually a ratiometric photoacoustic response, and that's the one we'll be demoing later this afternoon. What they all have in common, though, is porphyrin. All we do in our lab is based on porphyrin, and we use porphyrin because it is an endogenous absorber, and we can create systems that mimic nature. So all of these systems that I'm going to talk about have varying levels of biomimicry involved. So this is porphyrin. We all know what porphyrins are. They're heme and hemoglobin and steak, chlorophyll in plants, bacteria chlorophyll, and photosynthetic bacteria. So they've been used for a long time now in the clinic as photosensitizers. They absorb near-infrared absorption and are very potent photosensitizers. They also have near-infrared fluorescence. And because of their endogenous nature, they're enzymatically degradable. They do have some sort of inherent tumor affinity as well. And because of their biological functions in transporting oxygen and light harvesting, they can chelate metals. So we can do things like PET and MRI. So we're focused in our lab on creating technologies where we can take advantage of all of these different optical imaging, medical imaging, and then therapeutic capabilities in a single platform. So the first strategy that we used to create a photoacoustic probe, this is our flagship nanoparticle. It's called the Porphysome, out of porphyrin and liposome. So what we did is we take the porphyrin from algae. You can actually buy spirulina algae and just eat it as a supplement. And we conjugate it to a lipid. And so that conjugation then takes the very hydrophobic porphyrin and makes it behave more like a lipid. And so we can assemble it into classical nanomedicines, such as liposomes. And within each liposome, there's about 80,000 different porphyrin molecules. And so there's a very high density packing. This causes intense quenching. We are over 98% quenched, which makes them very efficient photothermal generators, close to gold, and of course, photoacoustic contrast agents. And what's really exciting for us about the porphysome is that we maintain this in serum as well, so we can maintain the photoacoustic and photothermal capabilities in vivo. So one of the first things that we did with porphysome was not for any optical application. It was, of course, for medical imaging. And so we were looking at prostate cancer. Prostate cancer is a disease that can really benefit from focal therapies and any agents that can provide delineation between the tumor and surrounding healthy tissue to aid in focal therapy and disease management. So we radiolabeled the porphysomes with copper-64, and we can see in an orthotopic prostate tumor model in a mouse, as well as in a metastatic model that has bone micrometastases, quite high depositions of porphyrin within the diseased lesions. Not only is there high deposition, there's also great contrast. So the tumor to healthy tissue ratio is very high. And so this really allows us to be able to delineate tissues. And what I would like to point out is that for PET, we're tracking all of the porphyrin. So whether it's in the nanoparticle or not, and then if we look at fluorescence, this is the recovery of porphyrin monomers after the porphysomes are dissociated within the tumor tissue and cells. And so we see that that contrast is maintained. So what we did next is we looked at photothermal therapy under MR guidance. And using the porphysome to heat the tissue, we reached ablative temperatures within about a minute. So this data set really gives us a lot of hope for some clinical applications. Three main ones. First, we can use radiolabeled porphysomes as PET imaging agents to both identify disease and monitor recurrence. And then we can use photoacoustic imaging to delineate tissues using porphysomes as the contrast agent. And then use photoacoustics as well as a measure for dosimetry before we do things like photothermal therapy. So really what we're creating here is a platform that requires advances in photoacoustic technologies. So we need a transrectal photoacoustic probe in order to do this sort of study in humans. So what we've done is we've created this whole network of researchers at the Princess Margaret Cancer Center, Sunnybrook Hospital, and it's focused on the main goal of getting the porphysome to the clinic and creating transrectal photoacoustic probe, and then merging the two in a human clinical setting. So right now where we're at for the porphysome side, which is our lab's main focus, we've mainly completed the preclinical phase. We've shown porphysome accumulation in multiple different tumor models. We focus mainly on prostate, head and neck, lung, and esophageal cancers. And then we've also done different animals as well. So we have mice, rats, rabbits, hamsters, and now we're into dogs. So we're now moving into the regulatory phase. We have scaled up to human scale doses, an ability to generate both porphyrin lipid and porphysomes. We're near the end of GMP, and we're nearing the end of toxicity studies in dogs as well. So we're hoping to soon move into the clinic. So you may have noticed I didn't show any photoacoustic data for the porphysome, and it's for a number of reasons. So we haven't had much success with photoacoustic imaging with the porphysomes. Two reasons. One, the agent actually absorbs at 670, not 680, so we don't get the peak. We have very low sensitivity because of that. So we're really waiting on technology that we can use specifically with the porphysome. It's also not a great platform for us to start moving into photoacoustics as well, because it's an intensimetric photoacoustic response and it's a turn-off response. So if we're going to start getting into the photoacoustic arena, what we need to do is shift the photoacoustic signal over and also create some sort of ratiometric signal. So this leads me to strategy 2. How do we shift a porphyrin absorption spectrum? So we looked at nature and specifically photosynthetic bacteria. So this is a electron micrograph of Rhodobacter spheroides. It's a purple sulfur bacteria. They live normally in hot springs, that kind of area. And each of these little circles is the light harvesting organelle. And if you zoom in on one of those, you can see the membrane is just covered with these light harvesting complexes. And these complexes are very highly regulated organizations of proteins and lipids that control the organization of pigments, specifically bacterial chlorophyll, which is a type of reduced porphyrin. And the way that it's set up is that light is absorbed at higher energies at the outside and then funneled into the reaction center at the core. And so this is depicted on the bottom left here. You can see all the different bacterial chlorophyll molecules in green. They're arranged in these rings. So what bacteria do is, instead of having many different types of bacterial chlorophylls that absorb at different wavelengths, they take a very limited set of bacterial chlorophyll dyes and change their arrangement to shift their spectrum and collect light of different energies. And this arrangement, this cyclical arrangement, lines up the dyes in a head-to-tail manner, and this causes what's called J aggregation. It's a type of exciton coupling. I won't go into the details of it, but all it does is it shifts the absorption. So you see for the dye, it absorbs at 750, and bacteria can shift that absorption all the way up to 875, just with a single dye. So what we did, of course, is take porphyrins. Instead of isolating them from algae, we isolated them from photosynthetic bacteria, conjugated it to a phospholipid, and recreated what nature has done. We're not as good as nature, though. We only shift from 750 to 824 nanometers. But if you look at the bottom right, that's a circular dichromosome spectra, and this gives evidence that what we're seeing in the absorption shift is really a function of structure. It's a function of the nanoscale arrangement of these dyes that creates this shift in the absorption spectrum. Within this structure, the dyes are mostly quenched, about 80% quenched. There is fluorescence, mainly due to the fact that we don't have a very high concentration of dyes in order to create this sort of structure. So what we did next is we took this dye and we did our standard intraoperative fluorescence-guided resection of sentinel lymph nodes. This is a rabbit model of head and neck cancer. So we did a peritumoral injection into the rabbit and then used the SPY system to actually dissect out the fluorescent positive lymph node. And then we took that lymph node and did photoacoustic imaging on it. We would, of course, have done this in vivo if the system was available for doing that. So this is still an intensimetric response, but there's something very, very exciting about the porphyrins that is unique to this system. So the structure that we're getting and the absorption properties that we're getting, it's structure dependent. It's nanostructure dependent. So the dye aggregation, it depends on its environment, and here we've put it into this artificial lipid membrane. So in order to have the dyes interact, they need to be in this very rigid lipid membrane environment. So if we heat that environment and we lose the rigidity of the lipid membrane, go beyond its transition temperature, we lose the J aggregation. So we actually turn off the signal at 824 and turn on the signal at 750. And it's a very sharp transition. We're using DSPC lipids, which have a transition temperature of 52.5 degrees. And from 52 to 53 degrees, we see the signal switch over. So this is what we'll be showing later this afternoon, so I won't give the photoacoustic data right now. So still, we have a number of disadvantages with the J-porphosome system. One is it's low loading, so we can only pack about 15 mole% into the shell before the whole particle just disintegrates. And so this gives incomplete fluorescence quenching. So from a photoacoustic perspective, we're losing a lot of signal that could otherwise be possible. And because it's also in this shell, it's sensitive to any serum interactions. So we actually have serum proteins that bind to the J porphosome and just burrow in and disrupt the aggregation. So what we wanted to create is something that is in the core of a particle, but that has a much higher density packing of dyes. So this leads me to strategy 3, which is a dual biomimetic system. So we take our idea of mimicking what nature has done in light harvesting complexes, and we merge it with another strategy used in nanomedicine development, and that's high-density lipoprotein mimetics. So we've now looked not at light harvesting complexes, but a different type of organelle in a different type of bacteria called green bacteria instead of purple bacteria, and these use what's called chlorosomes. So chlorosomes are really unique in nature in that they organize their dyes not by having this like rigid membrane of lipid and proteins. They do it purely by interactions between the dyes. So it's metal ligand interactions, hydrogen bonding, as well as this hydrophobic phytyl group that cause the dyes to form into these very large columns of dyes that have this J aggregation. So what we did is we created a synthetic analog of that, packed them all closely together within a lipid monolayer, and then took that and added an apoprotein mimetic. And so this formed this dual biomimetic system that looks like HDL from the outside and looks like chlorosome from the inside. And using the apoprotein actually provided a lot of advantages compared to just packing in the dyes together and adding a lipid coating. So if we just look at with the first biomimetic, you can see there is a shift in the absorption spectrum from 650 to about 710 nanometers, but it's not complete, and it's very easily disrupted by detergent. But if we switch over and add the peptide, now we have this complete shift in the absorption spectrum, and it's also very, very stable. It's actually really hard to disrupt these particles. So one advantage of this system is that it has very intense photoacoustic contrast compared to a lot of what we've created before because of this very dense dye packing. And you can even see that in the TM images, how much smaller these particles are with this apoprotein mimetic structure. So we did photoacoustic imaging just in a subcutaneous model, and these are unmixed images, so we can see the probe along the outside, the rim of the tumor where it's oxygenated, and not in the deoxygenated avascular core. So what I hope I've shown is that we're trying to develop photoacoustic probes that are for the future of technologies. They're not necessarily something that we're looking at immediate applications for outside of the porphosome, but we're really trying to figure out ways that we can add photoacoustic contrast to different nanoparticle systems that we already have, and where we can start adding photoacoustics as a way to help along either our applications or, in the case of this dual biomimetic system, how we can use photoacoustics to characterize the system itself. And so I've shown we have our porphosome, we have our J aggregating porphosome, and then we have miniature versions of both of those systems that use the lipoprotein format, and then we have our dual biomimetic, which we also have a larger version of that as well. So we're trying to create a library where we can start to use these particles in the near future. So with that, I'd like to thank Gong, of course, all the students that have shown their work. All of our imaging was done at STAR with the Vivo 2100 system, and all of our nanoparticle development is done in a CFI-funded nanomedicine facility that we started. So thank you.